All right, today we're going to crack the code on a real scientific detective story. This one takes us deep inside our own bodies to uncover the secret life of one single, absolutely crucial molecule.
So let me ask you this. What if your blood pressure pill, the one so many people take, had a secret, super complicated link to Alzheimer's disease? I know, it sounds like something out of a medical thriller, right? But this is a real puzzle scientists have been wrestling with for years.
And at the very heart of this mystery is one of the most important molecules in our bodies. It's a real double-edged sword, or in this case, a double-edged enzyme. So, let's meet our main character, the angiotensin-converting enzyme. We just call it ACE for short. And look, you've almost definitely heard of ACE inhibitors, right? They're a go-to frontline treatment for heart disease. And ACE's main job, its whole claim to fame, is keeping our blood pressure in check.
But, and this is a big but, this is where things get really complicated. On one hand, ACE is a total hero. It controls blood pressure, helps treat heart failure, protects our kidneys, all good stuff. But it's also got this mysterious, kind of darker side. See, it also breaks down other things in the body, including those amyloid beta peptides that are so famously linked with Alzheimer's. And that makes its actions, complex and super unpredictable.
So what happens when you block ACE with those ACE inhibitors? Well, they come with a whole list of known side effects. That annoying dry cough, sometimes swelling, and there's even been a potential link to higher lung cancer rates. But the real head-scratcher, the part that's driven researchers crazy, is its effect on Alzheimer's. For some patients, blocking ACE might be good, but for others, it can actually be harmful. It's been a total coin toss, a medical mystery.
So you gotta ask, why, right? Why is this one enzyme so difficult to get a handle on? And for decades, the answer was actually pretty simple. Scientists were essentially working blind. They were missing the blueprint. I mean, they knew what ACE did, more or less, but they just couldn't see how it worked on a deep structural level. And that is absolutely everything when you're trying to design smarter drugs. So what was holding them back? This quote from the actual research paper really nails it. For years, scientists could only get these blurry, incomplete glimpses of the ACE enzyme. They could see individual domains, like little pieces of it, or partially denatured structures, which basically means broken pieces. It's like trying to figure out how a whole car engine works when all you have is a spark plug and a couple of bolts. It's just not enough. Yeah, they had some puzzle pieces, but they could never, ever see the full picture on the box. And without that complete, high-res blueprint of the entire machine, trying to design a precise drug that only targets one specific function, forget about it. It was pretty much impossible. But this is where the story gets good. This is where our scientific detectives finally get their big break. A new technology, a real game changer, finally let them see what was previously unseeable. And that game-changing tool is a technology called cryo-electron microscopy, or cryo-EM for short. This thing is amazing. It's an incredibly powerful microscope that lets scientists see the tiniest biological molecules, like down to the level of individual atoms. It's wild. Okay, so they have this amazing new tech, but of course, there was a catch. There's always a catch, right? To use cryo-EM, you have to flash freeze your sample to hold it perfectly still for the picture. The problem? The ACE molecule is super fragile, it's delicate, and it just kept falling apart during the freezing process. One part of it was way less stable than the rest, and it would just get mangled or denatured before they could get a clean shot. And wow, this really shows you the solution in action. The researchers came up with a new, much faster freezing method they called chameleon. Just look at the difference. On the left, that's the old way, a blurry, incomplete mess, right? But on the right, that's with chameleon. Crystal clear, you can see the whole thing. This new technique protected that fragile molecule, and boom, for the very first time, the complete shape was revealed. Okay, so this brings us to the climax of our whole detective story, the big reveal. Not just what ACE actually looks like, but, and this is the crucial part, how it moves. And there it is. Take a look. For the first time ever, we are seeing the complete, high-resolution 3D structure of the human ACE enzyme. It's what's called a dimer. That just means two identical protein chains have teamed up. And you can see all four of its active parts, the so-called catalytic domains, all perfectly intact. This is the missing blueprint. This is what scientists have been dreaming of for decades. But here's the thing. The real aha moment, the mind-blowing discovery, wasn't just this static image. It was realizing that ACE isn't just a fixed object at all. No, it's a dynamic, moving machine. It's constantly in motion, constantly shape-shifting. In fact, the researchers managed to capture it in several different states. A great way to think about it is like a pair of molecular jaws. They saw it in an open state, with its jaws wide open, ready to chomp down on large peptides. They saw it in an intermediate state and a closed state, where those active sites are tucked away and hidden. The enzyme is literally always moving. And here you can actually see two of those snapshots side by side. It's incredible. The researchers basically caught the enzyme in the act of breathing, flexing and moving between these different functional shapes. And that dynamic nature, that's the secret. That's the key to understanding how it can do so many different things in the body. Okay, now this is where it gets really wild. This table kind of breaks down what they found. Just look at that top row for a second. In one single snapshot of the molecule, one part of the enzyme is open, while another part is closed at the exact same time. So it's not one big, uniform machine. Different parts can be doing different things all at once. This just unlocked a whole new level of understanding. It's way more complex than anyone thought. So let's bring it all back to our original problem. Now that we have the complete blueprint, what's the point? What can scientists actually do with this? Well, the answer is huge. They can finally start designing much, much smarter drugs. Think about the drugs we have now, the current ACE inhibitors. They're like a master key. You stick it in the lock, turn it, and it shuts down the whole building. It blocks the blood pressure function, which is what you want, but it also messes with all the enzyme's other jobs at the same time. And that's where all those unwanted side effects come from. But now that we know about all these different shapes and moving parts, we can start to imagine a totally new class of drugs. The paper calls them domain-specific modulators, which is a mouthful, I know, but a better way to think of them is as precision keys. Instead of one master key, imagine having a whole key ring, with each key designed to fit only one specific lock, a drug that targets just one shape of the enzyme to do just one job. And that leaves us with this final, really fascinating question. We finally have the complete blueprint. We know this enzyme is a dynamic shapeshifter. So is it possible? Could a future drug be designed to target just one of its many forms? Could we have a drug that lowers blood pressure without ever touching its role with Alzheimer's-related peptides, or maybe even the other way around? That right there is the incredible game-changing possibility that this new picture has finally unlocked.